Welcome to Mechanism of Disease. This is a presentation by Sean Haverson for Central New Mexico Community College. Mechanism of Disease will help prepare the EMT and paramedic to understand disease processes before understanding how to treat the disease process. This terms list is our objective list for definitions and concepts associated with this lecture. We will have this at the end of the lecture. It is also available for you in the learning module so that you can check your understanding and knowledge base. You can press pause if you need to go back to the learning module and print this off. As we can see, health is defined here as not just simply having the absence of a disease, but also being well-balanced and having a well-being. Something that I refer to throughout lectures and teaching convention and even in my thought process in treating patients is that health and disease are essentially imbalances in our homeostatic process. To make a well-rounded person in general health, we want to make sure that the physical, mental, and social well-being are always present, and an absence of one of these things or imbalance of one of these things will lead to disease. Etiology as a term is something that's going to be on your terms list, and etiology is a study of the cause. In fact, the part here, eti, is going to refer to a definition of cause. And I like to break down terms on occasion so that we can help relate what that term, when it's foreign to us, actually means. So the etiology is the study of the factors that are leading to the cause of disease. And there's a lot of different factors that are going to impact the health of an individual organism. We've got some more terms to review. We've got idiopathic as a term. And so, again, breaking this down, if we were to break down the term idio, and separate this. Idio by itself is going to refer to an idea that something is distinct, new, original, not associated with other things. So let's put distinct here. And then pathology as a term is going to refer to the disease. And that refers to pathos, which equals disease. So essentially, if somebody says that we've got an idiopathic disease, we have a disease that doesn't have a known or obvious cause to us. Some terms that you're probably aware of, signs and symptoms, but breaking those down into something a little bit further is using objective versus subjective review. An objective review is something that we can see and we can show to others and is very clear. So something that would be objective in a patient's process, a sign, it's a sign of something, would be vomiting. Vomiting is objective because we can see that person producing emesis. If we look at subjectivity, that would associate with a symptom. And so if we're looking at nausea versus vomiting, the nausea is going to be the symptom. We don't necessarily see that a patient is nauseous. That's something that they feel. But we can see the vomit and the emesis that's produced. Pathogenesis, as a term on your term list, is going to be the pattern of a disease's process. Again, pathos as a root cause, and so that's going to be the disease's development in the organism. More terminology. Morbidity, mortality, and prevalence rate are going to be terms that you're going to see specifically in scientific literature. And so comparing and contrasting morbidity versus mortality is important. So when we look at morbidity, morbidity comes from a term morbidus. Let's write that down. Morbidus. And morbidus means sickly. It's easy to get these two things confused. So something that is referred to as morbidity is not talking about the death of an individual, but the incidence of illness in a population. And we can define that population in many different ways. In fact, if we define the population very, very specifically during a time period, right? So maybe in 1960s, we're looking at a disease. That's a prevalence rate of how that illness is impacting the population. If we're looking at how many people die from the disease, that would be our mortality. And mortality makes me think of a mortician who's evaluating death. Well, let's look at the use of some of these terms. So if we were to look at cardiovascular disease, we would see that in 2010, so we have a time period here, so we are essentially reviewing a prevalence, but if we looked at the prevalence of cardiovascular disease in a subset population that's greater than or equal to 65 years of age, we would see that cardiovascular disease impacts 19.8% of that population. So really, we're looking at a morbidity. A global morbidity, well, less global, let's bring it to the national level. This is the morbidity, the rate of incidence of disease in the United States. So in 2010, 11.3% of the population, or 26.6 million people, were impacted by this. And let's put that into a framework. 26.6 million adults is really difficult to comprehend, but that would essentially be the same population if we combined in 2012 the population of New York City plus Dallas-Fort Worth combined. That entire population of people represents the number of people affected by this disease. If we look at the mortality rate of the disease, in 2010 the mortality rate, it was cardiovascular disease led to death in 193.6 people in a population of 100,000. So that's the application of our terms morbidity, mortality, and our prevalence rate. There's patterns that come up in disease, and they're the point of review by specific public health individuals, and that's to help identify any patterns that may be impacted or indicative of underlying disease transmission. So epidemiology is the study of those patterns and the occurrence in diseases in human populations. A term that might come up in epidemiological terms is endemic. So endemic is an outbreak of disease in a local region. So we could say perhaps if we're looking at Africa, maybe this is a tribe that's impacted by a specific disease. Epidemics would be the next step, and this is where a lot of folks, a population of people, are impacted by a disease at the same time. And epidemics can cover different geographical regions, but the idea is that all of these epidemics are tracking a disease that's outbreaking, and so it's happening very quickly as it progresses. Another term is the pandemic. Pandemics are widespread, and we generally talk about global epidemics. So we have a rapid growth of a disease that's happening over a large area. And so the pandemic that's come up in the last few years is associated with flus, and there's risks of multiple flus breaking out, and every few years a flu that might be impacting only fowl or other animals, non-humans, will change its structure and morph into a new disease that can now impact human beings, and sometimes it's very virulent. It translates through those populations really quickly. The impact of that can be great on populations. Let's look at this video. This is a video identifying the spread of an outbreak starting in Mexico. And so we're going to see that the outbreak will start in Mexico and branch out to the other regions of the United States, then transferring into regions of the world. And because of the air travel and other mechanisms in the world, this stuff travels very, very quickly, as we've seen in the past. I should note that this video is found on YouTube, and it's by Brockman Labs, specifically by Dirk Brockman, who takes information and translates that to mapping systems. If we look at pandemics, an example that happened in the early 1900s is the Spanish flu. Spanish influenza impacted 20 to 40% of the entire world population. All right, so if we're talking about the world population falling ill and not dying, this is essentially a morbidity rate. 50 million people died worldwide, so that's mortality. The spread of the Spanish flu occurred very, very quickly, and we can see here a time period. This is actually an original mortality diagram that was presented after the mortality had struck in 1918 and 1919. So early on, actually, this would be preceded by March of 1918. In March of 1918, there was a small outbreak at a U.S. Army camp. Small outbreak. A few soldiers were sick from this novel disease. It actually started, they think, in swine and translated to human beings. What's interesting, though, is by June and July, this had already spread to start impacting folks worldwide. So within a few months, we had a pandemic. The epidemic and endemic had started in the United States, but the pandemic rapidly started to impact the rest of the world, becoming a pandemic. And these rates were pretty staggering. So we can see here that these rates are going to be expressed as an annual rate per 1,000 as our capita, and we have variations here. What's interesting to note is that it did impact populations within the summer, but we can see that a pretty significant rate of death had increased during the October and November time period. And interesting to note that this was impacting generally healthy adults, not the populations of extreme, young and old, that we're used to seeing impacted by the flu deaths. More on mechanisms of disease. So we're now looking at pathophysiology. We've got some terms that we've talked about to help orient ourselves with what we would be reading and what we're learning about diseases as we move forward in our education and continuing education. Pathophysiology is the study of underlying physiological aspects of disease, and so that means that we're looking at how they impact the actual organ or tissues. And there's mechanisms that lead to the ability for these diseases to break out. We've got genetic mechanisms, pathogenic mechanisms, so those are things perhaps that might be spreading disease, genetic impact on disease, and then tumors and cancer. Looking at some of the mechanisms of disease in greater detail, we could have physical and chemical agents, autoimmunity, inflammation, and degeneration. And we'll look at a list of just a couple things that are associated with our physical and chemical agents here on the next slide. However, autoimmunity, inflammation, and degeneration also are mechanisms of disease. So if we're thinking of autoimmunity, perhaps the body's attacking itself. And so a disease that might remind us of autoimmunity is lupus, in which the body's cells are identified as invaders and the body attacks it. Inflammation can be associated with a number of different processes and exposure types, but we could think of something as simple as a person having a reaction to something floating around in the air, and it impacts their bronchi, and we have the onset of asthma. And then degeneration, this is where the body starts to break down as a mechanism of disease. And so as we get older, both male and females, although this impacts females more, this impacts more females than males. Degeneration might be exemplified by osteoporosis. Here is a list of physical and chemical agents, and there's so many different things. If you're looking at occupational medicine, noise and vibration are really big routes of disease that can impact a worker if they're exposed to these things without any protection. So you can imagine that OSHA would be very interested in decreasing noise levels or protecting from noise levels and vibration. And it's interesting, how would vibration impact a person? Well, if the person's working on machinery and the machinery doesn't have a lot of mechanisms to dissipate that energy, then the person might end up with, if they're riding the machinery all of their career, maybe a back injury. Maybe we've degraded some of the natural cushioning that's inside the spine, and the vertebrae are starting to rub up against each other. Kinetic energy is something that we deal with a lot in EMS, and that's in our trauma. Chemicals are all around us, and so chemicals as a route of exposure, as a route of disease, can come up through. And cigarette smoke, caustic substances, CO poisoning, obviously, can be a big problem. And then reactive oxygen. Reactive oxygen is actually thought to be a chemical that is impacting the rate of cancer in our tissues. Risk factors are identifying the things that may lay the ground for a disease to occur. just because somebody has one of these things or multiple of these things doesn't mean they automatically have the disease. It just means that they're likely at some point to get the disease given the presence of these conditions or predisposing conditions. And so genetic factors can tie in a lot of different things. They're generally things, genetic factors are things that are uncontrolled risk factors. We can't do a whole lot about a person's genetic factors so far. There's things that are coming onto the horizon, but usually the sex of a patient is going to determine these things. Maybe their heredity. Perhaps they're living in a group of people that has a higher incidence of disease because of heredity. Age can impact the disease process, young, old, and everything in between. Lifestyle, and I like to think of lifestyle and stress as coming together to produce a risk factor. Environmental factors are things that people are exposed to, and then our pre-existing conditions. And so those are all terms that we've heard before. Let's apply it to something here. So I'm going to give you just a moment. I'd like you to list examples of risk factors for coronary vascular disease, and in each of these categories. So list something that is genetic that impacts this or predisposes somebody: age, lifestyle, stress, environmental factors, and pre-existing conditions. I'll give you just a moment for that. Okay, you may still be working on it, but we just need one example, and let's keep your time on this to a minimum as we can, but let's look at some things that we might come up with. We know that for coronary vascular disease, genetic factors that are going to impact the person will be the sex of the person, right? We see this as something that happens more often in males than it does in females, but more and more it's really not a huge difference. How about different populations, different ethnic populations that are exposures? Age, if we remember tying genetic and age together, generally we see coronary vascular disease as a heightened risk at age 35 for our males and 45 generally for our female populations. Not exclusive though. That can occur at different times depending on other factors like lifestyle and stress. So tying these things together, obviously a stressful work environment, right, can decrease this. Lifestyle or increase the exposure. Lifestyle can be impacted by perhaps a lack of activity, maybe not working out, more sedentary lifestyle. Environmental factors, things that people are exposed to. So, I mean, we could break that down into chemical stuff, chemical factors, right? So chemicals, how about tobacco? How about lipids, right? Exposure to lipids. Pre-existing condition, one that comes to my mind is hypertension. Hypertension as a condition. Pathogenic organisms can be broken down in quite a few different types of organisms and particles as we go through. So we're just going to highlight this idea. We're not going to look at everything that you could be exposed to, but the core characteristics of each of these types of organisms. So a virus is generally a mechanism of disease that is going to invade your host cell and starts to take over those little tiny working factories inside of your cell to do what it wants rather than what the cell was originally designed for. So if we think of that term, it takes over a host cell to do something, it lives off the host cell, then this absolutely is going to be termed as a parasite. Now a lot of times they're very simple in structure. They have the genetic material that they need, and they have a protein coat. If we look at this process here, we can see just the size of a bunch of different viruses that are out there. And these things are really small. So if we're looking at things that are 0.5 microns, 0.5 microns is generally looking at the millionth in size. And so we can look at the millionth in size and kind of put that into relation to something. This right here is it's actually a mobile home that was designed by a sheikh in the United Arab Emirates. And the mobile home that's designed here is just about 42 feet in height. 42 feet. Now, if we were to compare that, 42 feet is essentially 1/millionth of the size of Earth. And the size of Earth is, if we're looking at diameter, 7,900 miles, essentially. So that's the scale that we're dealing with when we look at those cells. Here's how a virus might work. A virus is going to generally take over a host cell. So let's say this is a cell inside your body, right? This is a human cell. And the cell has stuff inside of it, those organelles, right? So we've got organelles. All right, maybe another one here. Yep, ribosomes would also be organelles, and there's others that are in there, but let's just kind of keep it simplified. So if we're looking at this virus here starting to attack the cell, the virus is going to make its way into the cell here on step 1, and then the genetic material is going to be transferred from the virus, and it invades the host cell. You can see here it's now part of the host cell. And that genetic material is going to take over and tell the cell to start making things. In this case, let's say that we want to make more viruses so we can spread them through the body to other cells. That's the goal in replication. So it's going to use the normal functioning process of the cell, but it takes it over by basically providing a new script, a new book that's going to define how these things and what these things are that are made. And so we've got proteins that are produced through the translation. The ribosomes are going to fold those and pleat those and make those useful. The Golgi apparatus might be a process that we use to produce some of those protein cells on the out portion of the cell, and that protein structures on the outer portion of the cell. And that makes this cell essentially now a replication factory for the virus. Or it could just basically create the virus and excrete it to the extracellular space, so now it can make its way through, find another cell to destroy, and attack it. This picture here is showing a T cell, and the T cell has been invaded by the HIV virus. And so on this picture, the red huge glob of the T cell has these little blue buds on it. All of those little blue buds are the buds that are new HIV viruses being excreted out of this cell, and they're going to make their way through the rest of the body and attack other cells. Now, the T cell is very useful in helping to battle infection, but because it's been taken over by the HIV virus, it's now essentially a zombie. It doesn't do its own work anymore, so it's not there to help against the invasion of other infections, opportunistic infections, as we know with HIV and AIDS. It only exists to create the HIV virus. So that's how this virus makes its way through the body very, very quickly, is the replication. You can see here there's likely thousands upon thousands of HIV excreted from this one cell. So we've got a question here for review. Which of the following is the result of a pathogenic virus? So we've got the influenza, anthrax, diarrhea, and histoplasmosis as examples. So what would be a virus? So you've probably heard it a few times. The influenza is a virus. So if you answered A, you would be correct. Moving on to other types of pathogenic organisms, we've got bacteria, and bacteria have a lot of different ways of being classified. Bacteria are small cells that lack a nucleus, but they are actually built to be pretty tough. As you know, bacteria can live on the surface of tables, for depending on the type of bacteria that we're exposed to, so do virus. Bacteria are living in very hot and acidic environments where other cells wouldn't be able to exist, and so they can actually hang out for a while and wait to infect the host cells that they're going to be exposed to. There's some classifications. One type of classification might be the lack of or the presence of oxygen in its ability to exist. And so aerobic versus anaerobic might tell us where we'll find this organism. Anaerobic organisms will likely be in a place where there's no oxygen because it can survive with that lack of oxygen. Another type of classification, which really doesn't mean a whole lot to EMS so much because we don't look at these things specifically, but if we're looking at classifications of antibiotics, sometimes the classification of the antibiotics is specific to the type of staining that was used to identify it. And when we say staining, we're talking about the staining of the individual cells so that we can look at them underneath a microscope. So gram-positive versus gram-negative staining would help identify the type of cell under a microscope. More classification. This we actually do utilize in emergency medicine quite often, and that's the shape and the size of the actual cells associated with the bacteria. And so we've got rod-shaped cells, bacilli, cocci, round shells, curved or spiral rods, and then very small bacteria that have the properties of being a parasite. Let's look at some examples, if we will. This picture here is a picture of a colony of bacteria that's growing under a microscope, so we can see that this is actually a gram-positive stain. And that's because the stain was taken up by the bacteria. Now, if we look at the structure, we look at the shape of these, this shape would be classified as a rod or bacilli. And so what we're looking at here is Bacillus anthracis, and this is the anthrax bacteria. Here's the effect of cutaneous anthrax on a person, and you can see the characteristic black ulcer identifies. Now, there's many different types of anthrax that can infect human beings. The most common is cutaneous, but the less common but highly infectious and can be deadly might be pulmonary anthrax, as it impacts the inside of our lungs. Here's another type of bacteria. So you can see we don't have the rod shape here, and this is using likely an electron microscope. As we look at this presentation, we can see that we have little circles, we have little spheres that are created, and so that as a round cell would be the cocci. And so what we're seeing here is MRSA in its colonization. All of this is MRSA growing on a slide for us. MRSA specifically is a very dangerous infection because MRSA is going to be methicillin resistant. Methicillin is a type of antibiotic used to treat specific strains of bacteria. Methicillin resistant Staphylococcus aureus. And so the staph infection can lead to something like this: before treatment, during treatment, after treatment. And the treatment we're talking about here is antibiotics, and then also the use of maggots. We don't use that in emergency medicine pre-hospital right now, but the use of maggots to destroy the dead tissue, which is actually infectious at that point in time. So just some examples of bacteria. Another type of pathogen that is present in infectious process for human beings is the fungi. And so fungi can make up a few different types of classifications. We've got yeast, molds, and mycotic infections. But these can be some pretty difficult infections to treat, and they're all over the body. A lot of folks are going to think of fungi associated with the athlete's foot, perhaps. But there's other types. There's fungal infections that are going to occur inside the lungs. There's a fungal type of pneumonia. Here's a picture of the Candida infection, and specifically here we're looking at something called thrush, which is in the mouth. So you can see this cheesy color that's present on the posterior section of the oral cavity is actually the oral candidiasis infection known as thrush. Moving forward in our pathogenic organisms, we have protozoa, which are one-celled organisms that have an organized nuclei. They generally are going to be parasitic. Here are some types of organisms under the protozoa. We've got some groups associated with it. We have amoebas, flagellates, ciliates, and sporozoa. I'm not going to go through all of these. As you can see, there's terms associated with it that you can read. There's a process of possession of a flagella that's going to help to move this thing through generally fluid. Cilia helping to create waves so that it can move it through fluid. But let's look at an example of amoeba. This is an amoeba, and this is similar to the amoeba that would cause amoebic dysentery. And dysentery is an infection, generally the GI tract, that's going to lead to profound infectious diarrhea. And the diarrhea can be so profound that the death is associated with dehydration and obviously pH changes in the blood. They can move, but they don't necessarily have any feet. But these projections that we see here are feet-like projections. These here, feet-like projections that can help the amoeba to essentially crawl along, if you will. Pathogenic animals are another type, generally a parasite, that can infect human beings. And They're pretty complex. They're small, but they're pretty complex organisms. They're not unicellular. They're multiple cells going to the growth of this organism. And we can see here, this is a picture of a fluke. And this fluke is not present generally in the United States, but is very infectious. And let's see, let me give a stab at trying to call this, or give this. Let me give a stab at the name here. Schistosomiasis is what I'm going to go with. So you can see this has a portion of it that's going to be used for feeding, and so the mouth here, it's got a reproductive organ, and this could be carried on the surface of snails and infected in humans if they're touching a snail and they don't wash their hands, or if they're ingesting snails that aren't properly cooked. But this is a tropical disease specifically that kills 200 million people worldwide. 200 million people worldwide. We've got other types of animals, pathogenic animals, roundworms and flatworms. We just looked at the fluke. Arthropods are also infectious or can carry as a route of infection, which we would term as vectors. And so vectors are living organisms that transfer disease to human beings. And so there's a few. This is a slide from the World Health Organization and their campaign to help decrease exposure by vectors. So mosquitoes, sandflies, ticks can be common vectors. And just by themselves, parasitic infections, there are so many parasitic infections, hundreds and hundreds of types of infections that are carried by animals, vectors, and parasites. So here's another review question for us. The liver fluke, so it's a fluke that is going to infest and grow inside the liver, is a result of which of these? A pathogenic protozoan, a pathogenic virus, a pathogenic bacterium, or pathogenic animal? Our correct answer here is a pathogenic animal, and we just saw that in the slide previous as we looked at flukes. Prevention and control of these diseases that we've talked about. There's mechanisms of transmission. You can see picking your nose is obviously not a good idea. And one of the greatest achievements of public health in decreasing diseases was the eradication, or essentially the education that decreased the amount of spitting that was going on in communities. And so we've got a few different mechanisms of transition. We've got person-to-person contact, which if we can identify the way something is transmitted, we can identify the way things are transmitted, we can identify a means of decreasing that transmission rate. And there's also environmental contact, and so sanitation practices can help decrease that. Other mechanisms of infection include opportunistic invasion. And so a good example of an opportunistic invasion is a bacteria that's growing somewhere waiting for entry into your body. And so you think of healthcare workers cleaning their hands all the time. If they're washing their hands very frequently or using the alcohol without any type of lotion or rehydration, there's microfractures that are going to occur in the tissue of the skin. And that allows a means of the disease process getting in, or bacteria that's living on your skin or populated on your skin getting into your body and causing an infection. Now, transmission by vectors, we've used that term already. Vectors are usually a pathway of infection, and vector control is usually talking about pest eradication to decrease that transmission of disease. Other strategies, we might want to prevent some activity of these diseases. Vaccination is going to stimulate an immunity in your body, and you basically get exposed to this disease beforehand in a small, controlled fashion, so that your body can build up an immune process to defeat it if it sees it again in the future. Now, chemicals can also be utilized to destroy these pathogens, and so bacteria can be destroyed by antibiotics. There are antivirals and synthetic compounds that are useful in the treatment of disease. There's other types, specifically when we're looking at terms of decreasing disease, and so we've got sterilization, disinfection, and antisepsis. And you probably know these terms or have utilized something that has gone through a sterilization process in emergency medicine, and those are things that are pre-packaged and sterile until opened in our pre-hospital environment. Disinfection and antisepsis. A note on the difference between disinfection versus antisepsis. Disinfection means something is already infected. and we're killing that infection. So disinfective spray is killing an infection, a population of some pathogen on a surface. Antisepsis is a pre-treatment. So this is going to prevent infection. Generally speaking, the sterilization process is going to be the process that will have the most wide and deep destruction of these pathogens, but that only occurs under high heat, high pressure, and we don't reproduce that in the field in any process. We just utilize equipment that might have gone through this process. So here's a review question. In an attempt to prevent the spread of many diseases, which strategy involves killing and disabling pathogens on surfaces before, we have a key word here, before they can spread to other people? Aseptic technique, septic technique, antibiotics, or antiviral agents? Aseptic techniques. Remember, that's the key, is it's before. Antibiotics and antivirals are going to be utilized after they've spread to the host organism, in this case, the human being. A few more slides before we wrap up, and we're just going to discuss the idea of inflammation. The inflammation process is something that can be helpful or can actually cause disease, as we saw in our mechanisms of disease in earlier slides. The inflammation process is going to reduce injury to tissues when it's used in a non-pathologic impact. Signs of the inflammation are really synonymous with signs of infection, right? So signs include redness, heat, swelling, and pain. And if we think of how this occurs, redness, heat, swelling, and pain, this is all associated with the edema that occurs in the inflammation process, where certain chemicals are going to make our blood cells leaky, right? So we're going to cause them to dilate, increase the blood volume, and we're going to allow that process to let white blood cells travel out of the bloodstream and into the site of infection. So because of that dilation in blood vessels, leaky blood vessels, and increase in blood volume, we get redness, right? We see blood closer to the skin, heat because core body temperature is being radiated, and then we've got swelling and pain, which are the remnants of that edema pushing up on tissue and the pain coming from the pushing of tissue up against our neurons. There's mediators that allow for this inflammation to occur. We've all heard of things like histamine, right? And at the very base of knowledge, our layman's knowledge, histamine, we probably have heard of things that are antihistamine. And so taking something for your slight allergic reaction or allergies, right, to pollen, using something like Benadryl is an antihistamine, so it's going to decrease the inflammation mediator present in the blood so that it decreases the impact on the inflammatory response. Remember that inflammation can be localized or systemic, and if it's systemic, we might be dealing with anaphylaxis. We'll wrap up with this review question here. Histamine, prostaglandins, and kinins are released from damaged tissue. Collectively, they are referred to as inflammatory exudate, inflammation mediators, immunosuppressive drugs, and inflammation response. Inflammation mediators. So those are those chemicals or cytokines that allow the inflammation process to occur. As we wrap up, here's some terms and concepts, the same list that we saw before. You should be able to go through your PowerPoint slide and any reading that's associated with this and identify the terms for all of these terms to be defined, and also to list out some of the concepts associated with mechanisms of disease, risk factors, types of pathogens, mechanisms of transmission, and finally prevention strategies. And this should wrap up the mechanism of disease process and lecture. Thank you for your time. If you're looking for photo credits for the photos that were presented and diagrams that were presented in this, you can find that in supplemental information in this module.